What I really talked about is a series of work being done under funding from Stand Up to Cancer and AACR that's involved 7 institutions, 65 investigators across multiple institutions, and our focus has been on cancer in women's cancers in particular, how to target an enzyme called PI3K, which stands for phosphoinositide 3 kinase. And that enzyme was discovered in my laboratory more than 25 years ago, and turns out to be one of the most mutated genes in cancer. Certainly in women's cancers, it's very frequently mutated, breast cancer, in particular endometrial cancers. And it's also amplified in ovarian cancers and other cervical cancers and other cancers. So we decided as a team to try to figure out how best to use PI3K inhibitors in those cancers. And in particular, although we're seeing responses with single agents, there's some patients who have remarkable responses to single agent drug inhibitors of PI3K. Most women do not show such dramatic responses. So we're trying to figure out how to use drug combinations that will work better than single agents, and also try to identify who are the patients who will respond to single agents. If we could understand that, then we could get those women the drug very quickly, and then figure out drug combinations that will work for other patients. We're doing clinical trials that are based on preclinical models we have, in which we either take a piece of the patient's tumor out of the patient and transplant it into a mouse, or we genetically engineer a mouse to have the same mutations as we see in the human disease. And then we test the drug as a single agent or in combination with other drugs that are already in the clinic to see which drug or drug combinations works best in which mutational background. Now, as we go forward with the clinical trials with the drug combinations that make most sense based on what we find in the preclinical studies, then from those patients we do full exome sequencing and something called RNA-seq on the tumors from every one of the patients. And the goal there is to retrospectively determine why this patient responded and why this patient didn't. So we go in and deeply interrogate all the molecular events going on in each of those tumors. One patient responds, the other doesn't, and try to figure out why did this person respond. Now, in other cases, we have patients who have dramatic responses, but then eventually become resistant to the drug. And there, we try to get a second biopsy at the time the tumor becomes resistant, and then compare the initial tumor that was responsive to the secondary tumor that became resistant, and ask, well, what were the molecular events there? What mutations occurred during that interval that caused the resistance? If we can learn that, then we can be smarter up front of anticipating the mechanism of escape and give a drug combination that will prevent that escape. So that's the science behind what we're trying to do in these trials. Well, one of the combinations we're very excited about is combining a PARP inhibitor. In fact, a drug recently recommended for approval here in Europe and in England called olaparib, made by AstraZeneca. Combining that drug with a PI3K inhibitor from Novartis, a compound called BKM 120. So we found in our preclinical models that for patients that have p53 mutations, which is a very common event in triple-negative breast cancer, and also have either BRCA mutations or p10 mutations, that for those particular subsets of patients, that drug combination, or certainly for that in our preclinical models, that those combinations indicate a very strong response to that drug combination. Now, olaparib alone works well in some of these same patients, But we find that combining the two together has a much more dramatic effect than either drug individually. And so we've now started a trial in the United States that has at multiple institutions that has now enrolled 70 patients, and we've seen a number of responses. And we're hoping now to go forward into a phase 2 trial where this will be opened in more hospitals and potentially in Europe as well. So we're very excited about the preliminary studies with that combination. We had one patient with a complete response. This was a triple-negative breast cancer patient, metastasis to the lung, and who had failed all other therapies. And we saw really a complete response. The lung metastasis completely disappeared, which is extremely rare at that late stage in disease. We're doing this not only in triple-negative breast cancer, but also in ovarian cancer, because we often see that triple-negative breast cancer and ovarian cancers have very similar mutational events. And in fact, in the case of germline BRCA mutations, those are the two diseases most frequently occur in women. So our models then that test the triple-negative breast cancer in a BRCA background, we think will be predictive of responses in ovarian. And that seems to be playing out in the clinic in that we're seeing responses in both types of cancers. We're entering an era that's really unprecedented in cancer research, where we have the tools to, and for a relatively small amount of money compared to the total expense of treating cancer, we can do full exome sequencing, even full genome sequencing, to determine really what's going on at the molecular level in the cancer. And I think in going forward in clinical trials, this will become standard, that we will do this for every single patient in the future. Right now, only a few trials do this, but the ones that do have the chance of learning, even from the failure. If the patient doesn't respond, you learn something as much as for the patient who does respond. And that's the way we should be doing clinical trials, where everyone who goes on to the trial, we learn something from their experience in the trial. Even if we can't benefit them, we're still going to learn something from their experience.